One of our big innovations, beyond getting the DNA motors to perform logic computations, is finding a way to convert that information into a simple output signal, motion or no motion, says Selma Piranay, an Emory University PhD candidate in chemistry, and first author of the paper. This signal can be read by anyone holding a cell phone equipped with an inexpensive magnifying attachment. Selma's breakthrough removes major roadblocks that stood in the way of making DNA computers useful and practical for a range of biomedical applications, says Khalid Salaita, senior author. The motors can sense chemical information in their environment, process that information, and then respond accordingly, mimicking some basic properties of living cells. Our DNA computers are essentially autonomous robots with sensing capabilities that determine whether they move or not. The motors can be programmed to respond to a specific pathogen or DNA sequence, making them a potential technology for medical testing and diagnostics. Another key advance is that each motor can operate independently, under different programs, while deployed as a group. That opens the door for a single massive array of the micron-sized motors to carry out a variety of tasks and perform motor-to-motor communication. The ability for the DNA motors to communicate with one another is a step towards producing the kind of complex, collective action generated by swarms of ants or bacteria, Salaita says. It could even lead to emergent properties. DNA nanotechnology takes advantage of the natural affinity for the DNA bases A, G, C and T to pair up with one another. By moving around the sequence of letters on synthetic strands of DNA, scientists can get the strands to bind together in ways that create different shapes and even build functioning machines. Its high speed allows a simple smartphone microscope to capture its motion through video. The motor's chassis is a micron-sized glass sphere. Hundreds of DNA strands, or legs, are allowed to bind to the sphere. These DNA legs are placed on a glass slide coated with the reactant RNA, the motor's fuel. As the legs bind and then release from the substrate, they keep guiding the sphere along. When Piranay joined the Salaita lab in 2018, she began working on a project to take the rolling motors to the next level by building in computer programming logic. It's a major goal in the biomedical field to take advantage of DNA for computation, Piranay says. I love the idea of using something that's innate in all of us to engineer new forms of technology. DNA is like a biological computer chip, storing vast amounts of information. The basic units of operation for DNA computation are short strands of synthetic DNA. Researchers can change the program of DNA by tweaking the sequences of AGTC on the strands. Unlike a hard, silicon chip, DNA-based computers and motors can function in water and other liquid environments, Salaita says. And one of the big challenges in fabricating silicon computer chips is trying to pack more data into an ever smaller footprint. DNA offers the potential to run many processing operations in parallel in a very small space. The density of operations you could run might even go to infinity. Synthetic DNA is also biocompatible and cheap to make. You can replicate DNA using enzymes, copying and pasting it as many times as you want, Salaita says. It's virtually free. Limitations remain, however, in the nascent field of DNA computation. A key hurdle is making the output of the computations easily readable. It also limits the signals that can be read to those present in the electromagnetic spectrum. Although trained as a chemist, Piranay began learning the basics of computer science and diving into bioengineering literature to try to overcome this hurdle. She came up with the idea of using a well-known reaction in bioengineering to perform the computation and pairing it with the motion of the rolling motors. The reaction, known as toehold-mediated strand displacement, occurs on duplex DNA, two complementary strands. The strands are tightly hugging one another except for one loose, floppy end of a strand, known as the toehold. The rolling motor can be programmed by coding it with duplex DNA that is complementary to a DNA target, a sequence of interest. The computer readout becomes simply, motion, or, no motion. When I first saw this concept work during an experiment, I made this really loud, excited sound, Pyrenees recalls. One of my colleagues came over and asked, "Are you okay? Nothing compares to seeing your idea come to life like that." That's a great moment. These two basic logic gates of motion, or no motion, can be strung together to build more complicated operations, mimicking how regular computer programs build on the logic gates of 0 or 1. Pyrenees took the project even further by finding a way to pack many diff. She simply varied the size and materials of the microscopic spheres that form the chassis for the DNA-based rolling motors. For instance, the spheres can range from 3 to 5 microns in diameter and be made of either silica or polystyrene. Each alteration provides slightly different optical properties that can be distinguished through a cell phone microscope. They are exploring the potential for the use of the DNA computing technology for home diagnostics of COVID-19 and other disease biomarkers. Developing devices for biomedical applications is especially rewarding because it's a chance to make a big impact in people's lives, Pyrenees says. Thanks for watching. Please subscribe my channel for more videos.